Assalamualaikum and good morning to Madam Jannah. My name is ADIL bin Muhammad Anwar. My matric number is 23DBT20F1016. So I'm from class DBT 4B. So now I'm going to do a simulation of Meiosis 1 and Meiosis 2. So as you can see here is the prophase 1. During prophase 1, the chromosomes undergo what is called synapsis. So what is happening is chromosome 1 from dad and one chromosome from mom are coming together and they are wrapping each other really tightly. So what is really going on is that they are swapping part of their chromosome. In other words, these are pretty much identical except for the changes in their gene, and so they undergo what is called crossing over. So segments of chromosome from mom are switching with chromosome with dad, and same thing with chromosome 2 here. So now is that giving us, it's giving a variation. So what we are doing in crossing over is we are combining two chromosomes that we got from our two parents and making a brand new chromosome. So if you keep watching, what happening next is we then go into a metaphase 1. So as you can see that they are all lining up or meeting in the middle of the cell at what's called metaphase plate, and then now they could have organized themselves like this, with the blue one on the left and the green one on the right. So but they could have easily organized themselves like this. So they could have been a different position. So this will be a totally different orientation of the chromosomes. It also could organize like this from chromosome 2, or it could organize like this. And so what do we have? We have four different ways, like this, and this. So we have four different ways that these chromosomes could orient themselves independently at metaphase 1. So what is that giving us? Well, it's giving us a variation. So another thing that happens in metaphase 1 is that spindle is going to attach. So you could see that the centrosomes move to either side of the cell, and the spindle attached to the centromere of each of those homologous chromosomes. So during anaphase 1, it's pulling them apart. So we can see those homologous chromosomes now are going to either cell. And then during telophase 1, what is happening is we are reforming a new nuclei at each side. So we have divided the nuclei, which is meiosis, but now we have divided rest of the cell, and that is called cytokinesis. And so we are done with meiosis 1. So there are 2. So what is going next is we are going to go through meiosis 2. So firstly is prophase 2. Now during prophase 2, there is no more crossing over, but what is going to happen is those two chromosomes are going to line up again, and they are going to meet right in the middle. So and then the spindle is going to attach to each centromeres. And now if we look at what happens, each of those centromeres are being pulled to another side during anaphase 2. So I'm going to pull to another side. both have a crossing over here. And then finally, during telophase 2 and cytokinesis, we have created those 4 cells that we wanted in the beginning. So as you can see here, the meiosis 2 is successfully created.